Hello. Okay, gonna go through B2 biology, the required practicals for the B2. How do I turn this down? Okay, cool. So, hold on. Right. Yeah. This is 1 second. Let me tell you whether these practicals are coming up in Edexcel. Give me a second. Are you combined or triple? Um, yeah, investigating ecosystems. Yeah, so this is Edexcel B2 as well. Yeah, so this is Edexcel B2 as well. Yeah, and this first bit I'm going through anyway will be in Edexcel B2. Let me just see. Um, where did you do nervous system? Okay, the first part will be in yeah, just practicals today. The first part will be B2 is also in Edexcel but is in AQA as well, which is investigating the population. So, does anyone know how you investigate the population of daisies in the field? Anyone got something they can say? How would you investigate? So this comes up in ecology, which is in your biology paper 2 exam, and basically you could be asked two questions. One of them is how to investigate the population, and the key word here when you're doing this core practical is to check out this word. It's the first thing you need to spot, is whether they use the word population, and if they use population, we do the practical a certain way, and if they use another word which we'll look at after this called distribution, then we're going to do the practical a different way. 9 times out of 10 so far they've asked for population, but let's look at it. Yeah, we're going to use quadrats. That's absolutely right. Now we're not going to use the belt transect. We use the transect for something called distribution, which is the second one we're going to look at. We're going to use the quadrat here, and that is for investigating population. So if they say population, we're going to use a slightly different method. Yeah, distribution would be using the belt transect. We're going to do distribution after this as well. So there's two versions of this that can come up. So how do you investigate the population of daisies in a field? First of all, question is, why not just count all the daisies in the field? So why not just count the daisies in the field? If I want to find out how many daisies are in the field, why don't we just count them? Sorry, I just realised that's all under my photo there. Let's move those there. That would help. Oh yeah, you can see that. Cool. Yeah, too many daisies. Going to take way too long. Too time consuming. Right. So we've got a keyword here. Yikes. Because they could give you some messed up variation of this. They could give you some messed up variation where it's nothing to do with daisies. All right, they haven't done that, but you've always got to be prepared for the unknown. So it could be bacteria on an agar plate. Why are you not going to bother counting all of them? So we're looking at daisies in a field. It would take too long to count them all. Okay, so what have we got to do instead? What are we going to take instead? Instead of counting all of them, what ones are we going to count? Anyone know what's the key word? Begins with S. We're going to take a sample. That's right, Miriam. Yeah. So we're going to take a sample. Now, not a smaple, a sample. Don't know what a smaple is. So we're going to take a sample. Now, if you're doing the AQA syllabus, there's only two. Well, there's really only two main core practicals that ever come up in the biology paper 2, and that is this practical that we're doing right now and the ruler drop practical, which we'll look at later. So But if you're doing Edexcel, you don't need the ruler drop practical. So we'll look at that later. Right, we're going to take a sample. How are we going to take our sample? How are we going to make it representative of the field? What are we going to use? Anyone got an idea? I want to sample an area of the field. How should I choose what area? Should I go to the nice sunny dry spot? Okay, a transect we're not going to use here when we're investigating population. It's a good keyword. We're going to use, yeah, we want random. That's a really good word. So we're going to say randomly place. Let me just make this a little bit smaller. Hey, we're 75%. Okay. Okay. So let's number this a little. What are we going to randomly place, guys? Step 1. Well done for saying random. Yeah, Claire also added use a random number generator. Does anyone know where you find that? Yeah, Quadrat. Okay, so let's get to that. Randomly place, well done, a Quadrat. Now this is the moment you know, when you're saying this, it's not a transect. When I do a transect, which we're going to look at later, you're going to do a line across the field. You're not doing it random. You're placing the Quadrat on the rope or on the tape measure. That's not random. You're walking in a straight line where the rope is. You know where you're going next. When you're investigating population, you don't know where you're going next. So you're going to do it totally randomly. So randomly place a Quadrat. Now, if you want, and you've got a 6 marker on this, depending on how big it is, you might want to explain how you're going to do that. So if you want to know how to make it random, some of you have said use a random number generator, right? And that is found on your calculator. You are allowed in the exam to say shut your eyes and throw it randomly. Okay, that is still a way of generating where to put the Quadrat. You can say pull a number out of a hat, but what has a number got to do with where you put the Quadrat? So I'm using a random number generator and I get the number 6 on my calculator. How do I know where I'm going to place it? Just because you've randomly generated a number, how do we know where to place it? Yeah, so coordinates. So what you're going to need to do is kind of draw a map of the field and number it. So I doubt you're going to have to write this, but it's good to know just in case. Draw a grid of field, split into coordinates, then randomly generate a number where to place Quadrat. Now just in case they go into depth, like how would you randomly decide where the Quadrat's going to go, 3-mark question, then you can say, look, draw a grid of the field, split it into squares or a map of the field, number them 1 to 20 or however many squares go in the field, and then generate a number and go and place the Quadrat there. So most of the time you're just going to have to say place the Quadrat randomly, and then you're not going to need to go into that much depth. Okay. So, what next? So randomly place a Quadrat. Right, my Quadrat, got one here. That's a Quadrat. That's what one looks like. Looks like I'm in prison, but that's a Quadrat. I've put my Quadrat down. What do I do next? Now my Quadrat is down. What should I do next? What do you think? What do we do with the Quadrat next? We've placed it on the ground at box number 7. What should we do? Count the daisies. Awesome. I know it sounds really obvious, but if you don't write count the daisies, you're going to lose a mark. So now potentially, there is a mark for count the daisies, identify the daisies. So you could even explain, I've lost my key there. You could even explain how you're going to identify the daisies. What you guys now in the modern world, how could you identify what a daisy is? You're not expected to know what a daisy is. How could you find it out? Yeah, you could Google it, look on your mobile phone, sometimes people have something called a key card which has different flowers on it or plants with the names. So they might ask you these kind of questions. So I would say count and identify the daisies, right? Another side question. So I'm just going to put a little side question up here. It's not part of our thing, but it could come up. So let's just, oh, what have I done there? Why can't I change the color of the font? Never mind. Right. Extra. What do you do if you are okay? I've done daisies. That's fairly easy to count. What if I'm saying go and count grass or moss? What if you've got to count grass or moss? How are you going to count that? How are you going to count blades of grass? There could be 1,000 in one quadrat. Mass. Okay, then you've got to pick them all up and weigh them. So we don't do mass. What we do in this, you may have seen quadrats before. I know I'm getting excited and holding up a quadrat. The reason why we've got these squares is they will say estimate the percentage cover. So if it looks like 50% are covered, 50% of these squares are covered with moss, you just say, right, 50% of that quadrat is moss, and you do percentage. So when it's a small plant, we need to estimate percentage cover. So for that, what have I done there? Okay, estimate percentage cover. You're never going to get it quite right. You kind of got to work out, you've got to add up the half squares to make a whole square. And sometimes they've even asked a question before, like, they've given you a quadrat and said count how many squares are covered but with the moss or with the grass, and it's been loads of half squares. You have to add them up. So estimate percentage cover, add up half squares, so to whole that's a really bad explanation, but basically combine two half squares to make a whole square when you do it. You could be asked that. It's come up before. Okay, still now I'm down to number 3. Okay, so it could be that you just want to say randomly place a quadrat, count the daisies. Step 3, how many times do you think we should do this? Yeah, similar to geography. So how many times do you think you should place the quadrat? Say it's a 100-meter field. My quadrat is a 1-meter quadrat. 5, good. Yeah, so you want to do minimum 5. Say, like, you could say repeat. I'm going to say 10 times just to be extra, but you can say 5. 5's generally on the mark scheme, the minimum you need to do it. So repeat 10 times, then find mean daisies in, yeah, one quadrat. Cool. That's going to make your test much more reliable, because you may have, and it's going to get rid of some error. No. Like, so Yoshi, this one is random. So we won't need to repeat it at each point, because if you've counted the daisies correctly and there's 7, there's just 7 there. This one is a random sampling. When we do the transect, where we put a rope or tape measure, then you could, I suppose, repeat it. But I mean, yeah, you don't have to repeat it on each one, because not with like the quadrat. But if you do 10 spread out all over the field randomly, then calculate how many. So let's say now I know how many are in one quadrat. Let's say 7 is my mean daisies. So what do we do with my number for mean daisies? Let's say we've added all our quadrats up. Some had 10, some had 4, some had 3, some had 8. We found that the mean number of daisies is 7. We've got 7 daisies in one quadrat. How can I estimate how many are in the whole field? Brilliant. Times by the area of the field. So multiply by the area of the field. Okay, so that's your basic explanation for it all. I'm going to show you where it can get a little bit messy. So we're going to do, right, just go on this. So, right, speeding through, what do you need to know off by heart coming into your ecology? for the practical is randomly place a quadrat, count the daisies in the quadrat or whatever they're asking you to count, repeat 10 times, find the mean number of daisies in one quadrat, then multiply up by the size of the field. And that's really your basics. Now, if you wanted, you could go into depth on how you're going to randomize it and how you actually do that. You could say make a grid, numbers, pull a number out of a hat. So put pieces of paper in a hat, pull a number out. You could say use your calculator, random number generator. Or if you can't be bothered with any of that, you can say shut your eyes, spin around and throw the quadrat. Literally, as long as you've mentioned a method of randomizing it. Now, if you're counting something really small like grass, moss, lichen, which is the stuff that grows on trees or on rocks, then you're going to estimate percentage cover. Okay, and they may even give you a set of squares and ask you to estimate the percentage cover in like a separate question. Right, let's actually give you the calculation, because the calculation can be a little bit tricky. So let's have a go at the calculation. Yeah, this could definitely be a 6 marker. This is often a 6 marker. I've got a couple of examples, so we're going to do a couple of examples of a 6 marker for it. But first of all, let me just give you a disgusting calculation part. So if this was your field and you were trying to find the amount of daisies in the field, okay, so that's your field. I don't know why I'm bothering drawing daisies, but yeah, there you go. You got all your daisies in the field. You've slung round your quadrat. You've put your quadrats out in the field. Now you've found the average or the mean. So the mean number of daisies in the quadrat, oh my gosh, I can't spell, quadrat was 7. So you got 7 daisies in the quadrat. Next step, each quadrat is 0.25 meters squared. Alright guys, hold on, I'm going to give you some measurements. Estimate the amount of daisies in the field. Show your calculations. Right, yeah, this is definitely a very likely one and a very likely scenario. This has come up so many times in paper 2, it's unreal how many times it's come up. So it's very likely. Right, let's pretend our field is 10 meters by 20 meters. So guys, how many daisies have we got in the field? So what we want to do, say right, there's 7 daisies. Alright, I haven't spelt daisies right, but never mind, I've called them diases or something weird like that. So there's our details. Yeah, so the area of the field is 10 by 20. Well done. So you're going to pick up 1 mark for calculating the area of the field. What's the next step, guys? We know there's 7 daisies in a quadrat, and 1 quadrat is 0.25 meters. Well, if the area of the field is 10 times 20, that's 200 meters squared, right? Our field is 200 meters squared. How many quadrats can we fit in the 200 meters? 200 divided by 0.25 times 7. Absolutely. So, well done. So essentially, there's 4 quadrats in 1 meter squared. 4, because each quadrat is 0.25. So you could do 1 divided by 0.25 is 4. Or you could do 200 divided by 0.25. So let's have a little look. Okay. Yeah, so basically we've got 800 quadrats times 7. Too much maths, yeah. So there's 800 quadrats times can fit in that field. It's a 200 meter field. One quadrat is 0.25. 800 could go in the field, and we know that our estimate is that there's 7 in each quadrat. So we're going to do 800 times 7, and we're going to get 5,000. I just had to bust up my calculator there. 5,600. So cool. Yeah, the thing is, if maths isn't your strong point, there's going to be elements of the question, like describe how you would do it, and you get all the marks for that. So you can make up for the couple of marks you're going to lose on the maths. Remember, you're only looking at 70% on a question to walk away with a level 9. So you're looking at about 70%. So if you're walking away with 50% on a particular question, you've scored a level 7 on that question. So you've got to work it all out. You might drop marks, but then you might be like, hey, I can just calculate the area of the field. 10 times 20 is 200. You're going to score a mark for that. So you're going to get quite a few marks for this. This would be a 3-mark question. You get a mark for working out the area of the field, a mark for working out the number of quadrats, and then that could fit in the field, and then a mark for your final answer. Now, they could make it even harder, so I'm just going to hit you with one slight other difficulty that they can come up with in this, which is for your maths. So what if they say I don't know, what if they say you've got 100 no, let's not do that. Okay, just to make sure that you guys know what is 100 centimeters squared, how to convert centimeters into meters squared. I shouldn't really do 100 centimeters. What's the point in doing 100? I was just trying to think of what would be a good size of a quadrat. So some people make this mistake. Some people make the mistake of thinking 100 centimeters squared is equal to 1 meter squared. It's not. Just because 100 centimeters are in a meter, of course, when we're talking about a square meter, it's 100 times 100. So there's 10,000, 100 times 100 centimeters squared in 1 meter squared. So just remember that when you're converting centimeters squared to meters, you'll need to divide by 10,000. So if I give you 100 centimeters squared and say, ooh, what's the size of this quadrat in meters? You need to divide in meters squared, you need to divide by 10,000, not divide by 100 like you normally would. Okay, so yeah, so watch out for that. Yeah, watch out for conversions. And if you're doing cubed, 1 centimeter cubed into meters cubed, then there's a million. So like 1 meter cubed has 1,000,000 centimeters cubed in it, because it's 100 times 100 times 100. So just remember those conversions there can be a little bit tricky. Right, enough of my waffle. Let's move on to the belt transect. Everyone cool with this? How do you know you're doing this one? What word are we looking out for in the question? How do we know if we're supposed to do this whole random throw the quadrats, multiply, identify the daisies, multiply up by the size of the field? How do we know when we're doing this one? What word are we looking for that's in the question? Yeah, daisies, but they might say poppies. They might say grass, they might say moss, they might say mussels, not as in on people, but mussels like on the beach. Population, you're looking for this word, population. So when you get the ecology practical, if it says population, you're generally going to do this one where we randomly throw the quadrats out. We find out the mean per quadrat and then multiply up by the size of the field. Okay. Let's have a look at the other practical, the other version. So the other version is when they say this. The grass, so the grass is, one sec. If you're counting grass, okay, let me zing here. Okay, let's clear this. Okay, here's the grass. Someone says how much grass is in the field. And let's say this is the grass. Going to be pretty hard to count, right? This isn't a field, but this is just where I'm placing my quadrat. Let's imagine this is my quadrat and it randomly lands here. Are you going to count? Are you going to bother counting up all those blades of grass? There might be 150 blades of grass there. Are you really going to count all of those blades of grass? It's better to do this. Count how many squares are shaded in green and say, right, I'm going to say this is my quadrat here is made of 12 squares. I'm going to say 9 of them are covered in grass. So 9 divided by 12 times 100. My percentage cover for this one quadrat in this massive field would be 9 divided by 12 times 100. Now you can argue whether I've done whether all of those are fully, that might not be ultra accurate, because some people would say, hold on sir, that's only half a square. I should have really counted that as half a square. So but there we go, just giving you the idea of how to do it. You want to count ones that are only covered half as half squares and add them to other half squares. So yeah, but that's how you would essentially work out percentage cover within a quadrat. Okay, that's not a field I'm showing you. That's an individual quadrat within a field. Remember, the quadrat has these little squares for working out percentage cover. Let's move on to the next one. The next keyword you're looking for here is distribution. So, investigate the distribution of let's think of a plant, I don't know, poppies from the tree line to the water. Okay. Right, Claire, so let me zing back for a sec. So say this is the percentage of 1. It's a good point, Claire. So let me zing back to this one. Say I have another one. My quadrat goes here. All right. So then I sling my other quadrat here. This is only if you're doing something like grass. All right. And then I think I've just made this quadrat like twice as big. Okay. Whatever. I would then go, okay, this one is 15% cover. And I'm still going to do 10 and then work out the average percentage by adding all those 10 different percentages I get together, divide by 10 and get the mean percentage cover per quadrat. No, what you do is you just, instead of counting the number of daisies and saying there's 7 daisies, this time you're just giving your answer in there's 50% daisies in each quadrat. So you just find the average percentage in the quadrat. So here, what's the average between my two quadrats? I would go, if I only did 2 quadrats, which would be a rubbish investigation because it wouldn't be very accurate, I'd say 90 divided by 2. My average percentage of grass per quadrat is 45% because I've worked out the mean from those 2 quadrats. But you're going to do 10 and then work out the mean. And then, yes, Claire, you're going to multiply up by the size of the field to potentially, or actually, I don't think you even need to. You don't need to multiply up by the size of the field. You just say that's the percentage cover. You don't need to multiply up by the size of the field for that. When you're doing percentage cover, you can just say, right, once you know the mean percentage per quadrat, that's the mean cover for the whole field. You don't need to be asked to multiply up. Right, let's go back to this one. Investigate the distribution of poppies from the tree line to the water. So we're given 2 locations. What we're essentially asking you here is, do more poppies grow near the water or do less poppies grow near the water? Blah, blah, blah. So got your whatever. We've got poppies. They're all over the place like this. Now, this is where you're going to do, what we're going to do now, Well, first of all, we're going to say, they want distribution. They might not use the words. They might not use the word distribution. They might say, find out, do more poppies grow near the water? That means it's best to do this one, and this is the transect. So what you're going to do here, you're going to put a rope or a tape measure. I always think it's best to use a tape measure because you've got distance straight across. Now, there's actually, if you do A-level biology, there's different types of transects. But the moment you put a rope across, that's a transect. And anything that touches the rope, you can count, or you can do and put quadrats. And some people like to put the quadrats like that, next to each other, like that. Other people say, no, I'm not doing that. They do what's called an interrupted one, which is probably the best one to do. And you just place the quadrat here, then 1 meter, then another quadrat, then 1 meter, then another. So it's not random. So you mustn't say put the quadrat randomly. You are placing the quadrats exactly there and then counting up how many poppies are in each. And then we can plot a graph of distance to the river or the lake and number of quadrats and see, number of poppies, and then see, does it increase as we get closer to the lake? And then we can talk about abiotic factors, or abiotic factors like more water. The poppies need water for photosynthesis, so as we go closer to the lake, and we can give explanations. But to do the actual practical, you need to be able to write it up like this. Population would just be how many. So if they say how many are in the field, you know, population. Distribution will be like you're just trying to find out, do more grow in this particular location. But sometimes you can use either. Sometimes either work. So how do you do this one? You would say, oh geez, that's come out a bit big. Okay, there's 100 ways to say this, but it's basically a transect. Now you can say randomly pick a starting point, okay? So you can still use the word random if you really want, but you mustn't say place the quadrats randomly. What we're saying is I could start here, I could start my line here, I could start my line here, so I can randomly choose where I'm going to start. And I probably should do that randomly, otherwise it has something called bias. Like I might choose, oh, it's nicer and less wet over here, I'm not going to get so much mud on my new trainers. So that's bias. So you should randomly choose where to start. Randomly pick a starting point, then place rope. You can say, it's better to say tape measure because then it you get the distance as well. So randomly place, randomly pick a starting point. Oh, it's not letting me number it. It's being a... Okay. Place rope, tape measure across area. Place quadrat. So you're going to get a mark for just saying quadrat at regular intervals. By the way, when you're in exams, always give examples. It just helps you get marks. Like if you're going to say place the quadrat at irregular, just say every 50 centimeters or every meter. It's up to you. Every 2 meters, place a quadrat along the rope or along the tape measure. And then it's a case of the same marks from the last one. Identify and count the poppies. Now, I've made up the name daisies and poppies. It could be anything. So identify and count the poppies. And then we can say, draw a graph of results. And there you go. Draw a graph, draw a table, record your results. Yeah. And that's it for that one. This one you don't multiply up by the size of the field or anything. There's no calculation on multiplying up by the size of the field for this one. Cool. Let's have a go at a question. So I'm going to zing all the way back. And let's have a go at a question. No, we want to go here. No. Oh, there we go. I need to move my photograph. Oh, let's just shrink the camera. Boom, right. All right, guys. So let's have a go at this question then, guys. Some students estimated the population of daisies in a school field. So I'm already thinking I know which one they're talking about. They've mentioned the word population. This is the method used. Find a place where some daisies are growing, put the quadrat down. Does that sound right to you? Find a place where the daisies are growing, put the quadrat down. Count and record the number of daisies in the quadrat. Repeat the steps 1 to 3 at 4 different places in the field. Okay. Calculate the mean number of daisies per quadrat. Use the data to estimate. Sounds like a pretty good investigation, but there's a couple of things that they could have done better there. So tick 2 boxes. What could they do better? Use a random method to place the quadrats. Well done, Adam. Anyone else? It's 2 ticks for this one. So the other thing I would use a highlighter for, you don't want to come out your exam and go, oh, was there 2 things? What are 2 improvements to these guys' method? You're saying 1, do not put quadrats near trees. Repeat another 10 quadrats. Use a long tape measure. Use a random. They're investigating population. We don't really need a long tape measure. I agree with you for 4. Use the same person to place all quadrats. No, that's not important. Use a long tape measure. We're not doing a transect. How many quadrats did they do? They did 4 different places. Maybe it's repeat for another 10 quadrats. I would say repeat for another 10 quadrats. Okay, I like your idea, don't put them near the trees, but what does that mean? Does that mean it's random if we say we're not going to go near the trees? What have we done? Yeah, Claire, number 2. The moment we say don't go near the trees, that's our bias. Yeah, we don't want to go near the trees because it might be tree roots, might be hard to count the poppies or daisies or whatever it is. That's bias. So we're going to skip that. It's 2 and 4 your answers. Right, guys, 6 marker here. Ooh. Okay, let's check this out. This is the 6 marker. With an improved method, the students calculated the mean number of daisies. I'm going to write that so you can actually see that. They worked out the mean number of daisies as 7.65. The students use a quadrat, 50 centimeter times 50. So we're probably going to have to work that out in meters, which is going to be a bummer. The diagram shows the school field and the dimensions of the field. This is a seriously hard question. Calculate the population of daisies in the school field. Give your answer to 2 significant figures. Okay, I'm not expecting everyone to pull out 6 marks here. Right? That's a difficult one. They've given it, so we're going to get marks for calculating the area of the field, which they've not made very straight. We're going to get marks for our field is in meters, so we need to convert our quadrat to meters. So anyone be able to convert 50 centimeter times 50 centimeter to meters squared will get you a couple of marks. And then working out the area of the field and then dividing it by the area of the quadrat. So we need to calculate the area of the field, calculate the area of the quadrat, divide the 2, and then multiply by 7.65. Hold on, Adam. What did you... yeah, be careful, Adam. What did you divide? You worked out 2,500 centimeters are in the quadrat, but you divided by 100. There's 100 centimeters in a meter straight line. How many centimeters are in a meter square? So you worked out this 2,500 centimeters squared in the quadrat, because we did 50 times 50. How many centimeters are in a meter square? Just remember, a meter square is 100 centimeters long and 100 centimeters wide. And if you do 100 times 100 on your calculator, you get 10,000. So when I convert this into meters squared, I'm going to divide this by 10,000. And that's going to give me 0.25 meters squared. So watch out for that, guys. That's going to pick you up a couple of marks. We've just worked out the area of the quadrat. How are you going to work out the area of this field? I have no idea. So the area of the field is really tricky. Yeah, so you're going to have to split. This we know is 62 meters. So this is like in your physics when you're doing the area under the curve to find the distance traveled by a car of a velocity time graph. What's 62? So I want to calculate this rectangle first. 62 meters times 164. Anyone? Yep. So the rectangle is 62 times 164. Oh, jeez Louise. The rectangle is 10,168 meters. Right, so that's a separate mark. Now, we now know it's still 164 wide, but I'm going to do 130 because that whole side is 130 minus 62. I know that this bit is 68 because remember, they've got to equal 130. So I know that's 68, so I'm going to do 68 times 164, and then we want to divide by 2. That was a disgusting exam question. Yeah. I'm going to zing over to the answer. So 164 equals that divided by 2. Okay, but can you work out how many of these daisies are in the field? You've got to add these two together. Oh my God, this can't be right. Jeez. Yeah, I probably screwed this up. This just seems like too big a field. So that's the square meterage of the field. And then we're going to divide by 0.25 because that's how big our quadrat was. And then times 7.65, which is pretty brutal. Okay, that was pretty horrible. Yeah, that was disgusting. I'm sure I'm a little bit out there. Well, let's see what we've done there. That's 481. Well, I've run out of room. 766.4. Yeah, I'm definitely out because they wanted it to two significant figures. And there's no way that I've calculated that correctly. Okay, let's check the answer here, guys. Oh, yeah. Yeah, roughly, but I didn't put to 2 sig. So, yeah. You can see there, same thing. Now that needed to be to two significant figures, so rounded up to 4.8. That was a horrible, horrible question. Not nice at all. So 5-mark question that was, and not nice. You can always rewind and watch that back if you're like worried about how the hell was that. Here's another one. The students noticed a very uneven distribution of daisies in the field. Okay, so we've got a 6 marker here. This is quite disgusting, but this is actually nothing to do with the Quadrat experiment, but this is in your ecology. So explain how biotic and abiotic factors could have caused an uneven distribution of daisy plants. So yeah, they're talking about distribution, but they're not asking you to do the line translate. They're just saying, why would more daisies grow in one part of a field than another part of a field? 6 marks. And they basically talk about biotic factors and abiotic. So can you guys think of 3 biotic factors and 3 abiotic factors? Biotic factors are living things. So factors caused by living things that can affect a population. Yeah, so that are trees covering the sun. So the trees are biotic. But yes, that would mean you get less sunlight, which is abiotic. So that's how you can connect the two. The trees are alive, so that's a biotic factor, and they grow creating shade, meaning less sunlight, which is an abiotic because it's not a live factor, gets to the plants and they do less photosynthesis. You can also talk about pH level in the soil. Wind is abiotic. You're looking at water levels, abiotic. We'll have to go back and look at the diagram and see. What about the students? Are the students biotic or abiotic? If we go back and look at the school building, what would you call students? Biotic factor or people? Are they alive or not alive? And how could students affect the amount of daisies in the field? We've even got sports pitches there. How could that affect the amount of daisies in the field? Yeah, they could. They're biotic factors. They could trample the daisies and kill them. So students are living things as well. How much the caretaker waters it and waters the field, that's a abiotic factor because it's not alive, but water's needed for photosynthesis. You can link that to your science. Yeah, so anyway, let's zing because I want to just quickly get on to, there's like millions of answers here. You can rewind the video, pause it and check the answers so you can have a go at these questions at some point. Okay, this is another one. Water is an abiotic factor. Name one biotic factor that could affect the number of buttercups growing on a field. So what's a living factor that could affect the number of buttercups? Yeah. I will. I'll put, yeah. So you can pause this, by the way, and come back and rewatch. I'm just going to put the answers up. Describe a method to investigate if the amount of water in the soil affects the number of buttercups in the field. Right. This is a slightly different nuance. They've said the number of buttercups. So it is population. I would do a random 10 quadrats. Count up the buttercups. Find out how many are in one quadrat by dividing by 10. Work out the mean quadrat buttercups per quadrat. I'd also say I need to identify the buttercups using my phone, Google Images, or using a key card. And then I would calculate the size of the field, the area of the field. I would calculate the size of my quadrat, divide the area of the field by my quadrat and times by the mean number of buttercups, and that would give me the population of buttercups in the field. Now it says compare it if water affects the number. I'd find a wet field and a dry field and do the same in the wet field as I've just explained in the dry field, and then see which one had more buttercups. You probably can do a line transect here. You can say I'll find a field which is a water half a field, so it's wet, and do the other half of the field so it's dry, and then you could say I'll do the belt transect, and you could say place a tape measure across the field, place your quadrat at regular intervals, and that way you're getting the dry into the wet and seeing which one has more. But I would probably just do random quadrats in 2 separate fields. Anyway, you can pause the video when you rewatch and then check the answers. So here are the answers. Let me just blow them up in case. So these are the answers, guys. So yeah, this is the random quadrat. So one of them, they're giving you 6 marks for doing the Quadrat random, and they're actually saying you can do the line transect as well. So you could do the transect for the other 6 marker. So there's 2 methods. Right, I want to move on to the ruler drop. Yeah, you could do that with, yeah. Let's check out, I want to do the ruler drop prac. Alright guys. How are you guys with your ruler drop practical? So, ruler drop practical isn't too bad. We'll talk through some of the things. So, okay. Main thing with a ruler drop. What is the ruler drop practical? So often they do it like this one is with caffeine reaction times. You're testing people's reaction times. Essentially, the distance, the smaller the distance, I just caught that at like 10 centimeters. The shorter the distance, if I caught it at 3 centimeters, I've got quicker reactions. You get some rulers that they've got rid of the distance and they've put seconds, time, and they've basically done a math calculation and worked out exactly how much time that is equal to with the distance. But essentially, the lower the number, the quicker the reactions. Now you could take a group of people and give them caffeine or coffee and a group without, and you could find out, or you could do the same group of people without coffee and then with coffee. So you could test caffeine or other things on reaction time. You could do it when people are tired. You could give them a Lucozade drink, a Red Bull, whatever. Yeah, reaction times. The things you need to look out for. Your thumb and finger need to be the same distance each time, the catcher. Now when you're talking about this in a 6 marker, talk about it as the person who is dropping, call them the dropper. The person who is catching, call them the catcher. It makes it really easy to talk about. And so with the catcher, this reaction time one, there's loads of variables you need to control, and in biology you can never control them all. But keep the finger and thumb same distance apart. Have them catching with the same finger and thumb, so best to just use your index finger and your thumb, like that. We want 0 centimeters to be in line with the finger and thumb, but just make sure it's the same every time. What about my arm? What do we need to do with my arm? We need to rest it on the table, and it's best to be flat on the table so that I can't do this and change the position of my hand each time. So we need to control everything. Distance between the thumb and finger, rest your hand on the table. Now the dropper needs to hold the ruler in the exact same place each time at 0 centimeters. What about if the dropper lets go every time after 2 seconds? Is that good? So the dropper needs to randomly do it. And what about when you do this with your friend at school? You might start to read them. Like if you know your friend really well, you might be like, yeah, they're going to drop it now, boom, and start guessing when they're going to drop it. It might make your reactions look quicker. So it's really important that the dropper totally messes around with the timings and you have no clue when they're going to drop it. And obviously people get better and better at reaction times. Let's have a go at this question. Okay, this is a reaction time question. So let's go through and highlight some bits of it. Caffeine is a drug that affects reaction time. Doesn't tell us whether it increases or decreases it. Coffee is a drink that contains caffeine. 5 students, I'm liking that. It's a decent number. Investigate the effect of drinking coffee on their reaction time. Each student sat in front of a computer screen showing a reaction timer. This is the method used. Press any key on the keyboard when the color of the screen changes to green. Okay. Record the reaction time shown on the computer screen. Drink coffee. Wait 15 minutes. Repeat 1 and 2. Great. What is the dependent variable? No, you don't need to know the reaction time with the distance. You don't know how to have to convert it unless they give you a conversion equation. You don't have to know that. Just that if it's a smaller distance, they got quicker reactions. Okay, what's the dependent variable, guys, in this investigation? Is it coffee containing caffeine, number of students, or the reaction time? What is it that's the dependent variable? Yeah, you're recording the reaction time, absolutely. So the dependent variable is what you record, and that is the reaction time. Cool. Number 3. Right, 2 control variables the students should have used. That's right, Georgette, reactions, yeah. What are 2 things they should have controlled? You got 5 kids or students doing this experiment, clicking on the computer for their reactions. The computer's doing all the timing. It's much better than the crappy ruler drop. The computer's much better. There's loads of these free ones on Google. You just Google reaction time game and you can find out how quick you are. And it goes green and you tap it as soon as it says tap, and then yeah, it records your reaction time when it changes color. Yeah, we'll get to that. They will ask you that. Why is it better to use the computer instead of the ruler practical? And the computer is a lot more accurate at timing. There's no human error. The computer's not a human, so it can't mess up with any timings or anything. Give 2 control variables though. What are 2 things that? Yeah, what are 2 things they should keep the same about the students that might affect their reaction time? Yeah, the same people drinking the coffee. But what about the coffee? What about the coffee? They've just said people drink the coffee. Yeah, same amount of time. It said wait 15 minutes. What about the coffee? What haven't they said about the coffee here, guys? Or the caffeine? Same type of coffee. What about if one student decides to down a whole glass, another one just has a sip? Same amount, yeah, same volume, same amount, same concentration, same strength of coffee. Loads of things just on the coffee alone. Same strength of coffee, because someone might have put 1 spoon in one, 2 spoons in another. Same amount. When you're dealing with liquids, it's always better to say volume. But yeah, amount will normally get you the marks in GCSE as well. Yeah, and there's 100 of other things that could affect them as well, like, have they drunk a Red Bull earlier in the day? Like, did they have any other coffee in the day? What have they eaten? So all of these kind of things, with biology, there's about 500 things you could probably write there for control variables. Things like their age, if the age of the students should be the same. Why did the students wait 15 minutes after drinking the coffee before repeating the test? Why not just do it straight away? Down the coffee, have another go. Sorry. Thank you for that. There we go. Perfecto. Thanks for that comment about moving the camera. I would have just carried on. Okay, guys. Why did they wait 15 minutes? Very good, Adam. So it can get into the blood. Yeah, it needs to be digested, get into the blood. Good. Yeah, for it to have an effect. The drug's going to take time to have an effect. Good. Responding to the color change of the screen involves a receptor in the student. Where is the receptor? Where would you find the receptor when they're doing the color change? Yeah, absolutely. It's in the eye. So well done. Yeah, it's going to be your rods and cone cells in the eye. Great. And here's the answers. So the control variables, age, gender, time of day, amount of sleep they had, the health of the students, how heavy they are. Because if you're like 100 kilos, caffeine's not going to affect you as much as someone who's, same with like alcohol. If you're drinking alcohol, the bigger someone is, the less effect. The more cells they have in the body, the less effect on them. So yeah, all of that kind of stuff. They've got loads of things there, and there'll be loads more that aren't there as well. To give time for the coffee to be digested, time for it to work, time for it to have an effect. Absolutely. Right. Okay, here's a little bit more. Responding to the color change of the screen involves an effector in the student. What's the name of the effector? Yeah. I'll do hormones and menstrual cycle later in the week. What's the effector in the student? So you've got something called effectors with the nervous system. Yeah, the muscle. Well done, Claire. It's the muscle. So effectors can be hormones or glands or muscles. Glands release hormones, muscles contract. This one is contracting your muscle to hit the screen. What's the effect of drinking coffee on reaction time? Use the table above. So we can look here. What's the general effect on the reaction time? If we compare each student after they've drunk coffee, what's happened to their reactions? Faster or slower? Yeah, reaction time decreases. Awesome. Which student had the smallest change in reaction time? So you're looking for the one with the smallest change. You could probably just do this visually. That's about 130, 130. Looks like, anyone know which student? Yeah, student 3. Well done. There's only a change in 20 milliseconds. The students decided one result was anomalous. What should the students do with the anomalous result when calculating the mean change in reaction time? Student 1 had a 130 millisecond difference. Student 3 only had 20 millisecond difference. Which one looks like an odd result? Oops. Student 4 went from 871. Yeah, that was like, they were like 3 times faster afterwards. Everyone else was like under 2 times faster. It's difficult to say whether it's 4 or whether it's 3. There's not really been any change in the results in 3. Yeah. I don't like 3 and I don't like 4. 4 is like a serious big change in results. But you're right, before drinking coffee, their reaction time looks a little bit slow before drinking coffee. Probably a mistake on that one. What should you do? You should, when you get an odd result, whether it's 3 or 4, we'll find out in a second when we hit the answers. What should you do when you find an anomalous result? What should the students do? It actually, by the way, guys, it doesn't ask you for the anomalous result. That's not what it's asking you. So you won't get this wrong in this one. They're not actually asking you. They're saying, what should the students do with the anomalous result? Absolutely, Claire. Don't include it in the mean. Yeah, well done, Christian. Don't include it in the calculation. You subtract it and then divide by 4 instead of 5. So we don't use the anomalous result. Awesome. Well done, guys. Right, let's zing. Yeah, they don't even ask you what the anomalous result is. Probably because it was a little bit difficult to spot which one. This is just another reaction time one. I'm going to leave. I'm not going to go over this one. So what you guys can do, because I'm going to finish up, what you guys can do is pause the video, have a go. Not now, but you can rewatch it. You can pause, and I'll put the answers up. And this person is they're using stopwatches, and a student saying stop. Straight away, when humans are using stopwatches, that's going to be full of error. Yeah, by the time someone says, "Oh, wait, press stop," and you go boom, you're milliseconds out, whereas a computer is not going to be out, so with its reaction. So there's a lots of error there in pressing start and stop on a stopwatch, but all the answers are going to be up here, so you can have a go and then check. You can have a go, pause it, and then forward through and check the answers. So that's the answers for that slide. There's another one here. Again, they're saying they noticed pair 8 was anomalous. What did they do with their mean? I'm going to go live later in the week. I'm not sure yet. I'll put it up on. You can go on my channel, and I think there's probably a notification button, and it will notify you whenever I'm going live. I'm also going to release. I've got a few videos. I've got a few videos on the menstrual cycle and everything. So yeah, on my channel, I've got like AQA Biology Paper 2. I've got videos on the menstrual cycle with exam questions. I've got videos on other other topics as well. I haven't done a lot on B2, but I've got quite a few little videos where I just explain it and then give you some exam questions. But I am going to go live on and go over all of it. So yeah, you can pause the video and then check the answers. The answers are all here. And again, you can just you can rewatch it and pause it and check it off. So you can have a go on a scrap bit of paper. A lot of my videos have questions like this. Yeah, no problems. So again, this is a bit on the nervous system, which is one of the topics coming up in B2 if you're doing AQA. If you're doing EdXL, you've already done the nervous system in paper 1, I believe, so you won't have to do it again. So again, you can just pause the video and have a go with these, and then onto the old fields and these ones, which you can forward through and skip back and check these again. This is what the computer looks like if you go on Google and you want to find out your reaction time. Be careful, some of them just put ads and pop-ups on your computer, so it's not worth it. But basically, that's what a computer reaction time test looks like. It goes red, it says wait, 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 now. And you click your screen as fast as you can, or you hit any key on your keyboard as fast as you can, and it will tell you your reaction time. You can see here, that is the reaction time of a dead person. So, but I was just taking a shot to show you what happens. So it says wait, and then it says click me, and then when you click, it stops automatically. So the computer's doing the timing, and that takes out human error. There'll always be error when humans do timing, so it's much more effective to use a computer. All right, guys. In fact, computers, there's two reasons why computers are better. Less chance of, they don't have human error, so they're more accurate, but also they have higher resolution or more precise. Look how many milliseconds that goes to. That's like 13,161 milliseconds. So they're more precise as well. Okay. The other thing is, nice. Well done. The other thing with computers, you can't read the computer, can you? Like, say I'm the catcher and my mate's doing that, I can stare at them and I can watch and see if their muscle in their arm twitches or if they like pull a face or something. You can predict and guess and read the body language of a human person who you're doing a reaction time test with. You can't read the computer. You can't read its body language. So that's another thing that you can score a mark for in the exam. All right, guys, I'm going to finish the live there. I'm going to do some more in the week. So, yeah, I've got a playlist for B2 where I'm adding videos. I just did one today on diabetes type 1 and type 2 that's coming up. So you can watch that, and they all end with like an exam question. Unless they're like a 60-second short, then they don't have an exam question. All right, guys, take care. And have a good weekend, and I'll see you next time. Bye bye. Thanks, guys. Thanks a lot.